Hello dear viewers, myself Nasir and welcome to Mr.Zoologist YouTube channel. Today we are going to discuss about plasma membrane in detail. That plasma membrane is different from all the plasma membrane structures or details you might have learned earlier. That is the part of the advanced cell biology, and you are going to look upon the cell membrane or plasma membrane at the advanced level, master's or PhD level subject or topics you are going to look upon right now. The plasma membrane is a lipid bilayer, a flexible yet sturdy barrier that surrounds and contains the cytoplasm of a cell. It is the outermost boundary of the cell and is described by the fluid mosaic model. That is the most acceptable model for the structure of the plasma membrane. So in a fluid mosaic model, as you can see that most of the structure is embedded inside the plasma membrane and a very few structures are outside, such as like the proteins or some the signal receptors. That is the structure of the plasma membrane. As you can see on the top, these are the glycoproteins or carbohydrate protein complex, glycolipids. These are the complexes or conjugate molecules formed by carbohydrates and lipids, and these are the channel proteins, as you can see in the center of the plasma membrane or cell membrane. And these are the extracellular matrix with which cell membrane is bounded. So these are the two lipid bilayers. One is the upper and one is the lower. And inside is the cytosol of the cell. And these are the integral proteins. And that are the cholesterol molecules embedded in the cell membrane or plasma membrane. If we talk about the chemical composition of plasma membrane, the basic structural framework is the lipid bilayer made up of three macromolecules: lipids, proteins, carbohydrates. These make up the plasma membrane, and we are going to look upon them one by one in detail. If we talk about membrane lipids, that contain almost 75% phospholipids, 20% cholesterol, and 5% are the glycolipids. Mostly lipids are amphipathic, having both polar and non-polar parts. Means to say, they have one part that is charged, that means that is polar, and the other is non-polar, which means to say that does not contain poles or charges at all. So that acts as permeability barriers. Selective movement of the molecules is allowed by the permeability barriers and essential for the maintenance of fluidity of the membrane. And if we talk about phospholipids, as I have told you earlier, the head part is the polar part, and that contains the phosphate group PO4 negative, and the tail is containing the non-polar part, and that has long chain fatty acids. These fatty acids can be phosphatidylcholine, phosphatidylethanolamine, phosphatidylserine, phosphatidyl'inositol, plasmalogens, and sphingomyelins. These are all the fatty acids that are the part of the phospholipids. Cholesterol molecules are weakly amphipathic and are interspersed among the other lipids in both layers of the membrane. Interspersed means dispersed or separated inside the lipid bilayers. Glycolipids present only in the membrane layer that faces the extracellular fluid or matrix. If we talk about the fluidity of the membrane, the physical state of the lipid of a membrane is described by its fluidity or viscosity. Consider a simple artificial bilayer composed of phosphatidylcholine and phosphatidylethanolamine, whose fatty acids are largely unsaturated. You might be familiar with the unsaturation. If a compound is containing double bond or triple bond, that is unsaturated. And if a compound is containing single bond, it means to say single bond is a sign of saturation. So if the temperature of the bilayer is kept around 37 degrees Celsius, the lipid exists in a relatively fluid state. At this temperature, the lipid bilayer is best described as a two-dimensional liquid crystal, solid shape, or in the liquid indefinite shape. As in a crystal, the molecules still retain a specified orientation. Actually, crystal is linked with a definite shape. So the molecules still retain a specified orientation. In this case, the long axes of the molecules tend toward a parallel arrangement, yet individual phospholipids can rotate around their axis or move laterally within the plane of the bilayer if the temperature is slowly lowered. A point is reached where the bilayer distinctly changes, which is a prominent change you can see, or the visible change occurs in structure. These are the two types of the movements can be observed. Three types you can say that. First one is the rotational, rotates on its axis, on their place. Second is the lateral, position changing, and third one is a flip-flop movement from upside to downside, and from downside to upside. Movements can occur from upper membrane to lower and from lower to upper. That is a transverse or flip-flop movement. As you can see in the diagram, that in the first diagram, that is very slow, that time takes about half day, uncatalyzed transbilayer flip-flop division. First of all, that phospholipid was part of the upper layer, then it took place of the second or lower layer, that is the transverse or flip-flop diffusion. And next is uncatalyzed lateral diffusion. In lateral diffusion, one molecule was on the left side in the third place, and in the second diagram you can see that was on the second last place, and that has moved on the right side, and that movement was very fast, about taken time of 1 micrometer per second. So the lipid is converted from a liquid crystalline phase to a frozen crystalline gel, in which the movement of phospholipid fatty acid chain is greatly restricted, hindered, or weakened, or difficult to move. The temperature at which this change occurs is called the transition temperature. The transition is from liquid crystalline to a frozen crystalline gel to a solid structure. That temperature change is called the transition temperature. So here is in the detailed explanation of the flip-flop movement. So within a phospholipid bilayer membrane, the phospholipids and many proteins are in a constant state of lateral motion, changing their place from one place to the other place in the same membrane. That will be the lateral motion. Although lipids move at a constant state of about 1 micrometer per second, proteins move at varying speeds, and some are immobile. Not all proteins can move. This is because they are restricted by attachments to the other proteins, and they are also the part of the extracellular matrix. So that continues in the membranal structure of the plasma membrane. So phospholipids move relatively quickly along the lateral direction because there are not any energy barriers that must be overcome. On the other hand, what happens if phospholipid attempts to move along the vertical direction? This is called transverse or flip-flop direction or movement. Flip-flop, phospholipid flip-flop at a rate at which they move transversely is much lower. Means to say lateral diffusion is faster, whereas flip-flop diffusion is very much slower. That takes so much time due to the hindrance, due to the other energy carrying or barriers they have to overcome in order to flip-flop from upper membrane to the lower membrane. So that is the whole concept I have told you in brief way. So phospholipids move relatively freely and quickly along the lateral. Some but not all proteins can also move in a lateral fashion. Although phospholipids can flip-flop, they do so slowly. Proteins called flipoposes can assist in the transverse division. Proteins do not flip-flop. Only the phospholipids can do flip-flop movement. Nature of fatty acids. The transition temperature of a particular bilayer depends on the nature of the lipid molecules. Saturated fatty acids have the shape of a straight, flexible rod. Whereas unsaturated fatty acids, on the other hand, have grooves in the chain, breakage, or you can say a pore-like structure. Phospholipids with saturated chains packed together more tightly than those containing unsaturated chains. The greater the degree of unsaturation, the lower the temperature before the bilayer gels. The temperature will be colder. The introduction of one double bond lowers the melting temperature of the stearic acid almost about 60 degrees Celsius. The other factor that also affects the bilayer fluidity is the fatty acid chain length. The shorter fatty acid chains, the lower this melting point. The higher the fatty acid chain length, the higher is the melting point or temperature. As you can see in the first diagram, is in the cis fat containing the hydrogens on the same side. Opposite cross side, that will be the trans. Trans means opposite. Cis means same. So, role of cholesterol. The physical state of the membrane is affected by cholesterol because of their orientation within the bilayer. Cholesterol molecules disrupt the close packing of the fatty side chains and interfere their mobility. The presence of cholesterol tends to abolish sharp transition temperatures. Note there is rapid transition from the liquid crystalline gel to the frozen crystalline gel. Cholesterol stops this transition in the earliest if that occurs too much rapidly. So cholesterol tends to increase the durability while decreasing the permeability of the membrane. So what is membrane asymmetry? Plasma membrane has two leaflets, inner and outer. The leaflets differ in composition of the lipid and protein. Outer leaflet has phosphatidylcholine, sphingomyelin and glycolipids. Inner leaflet has phosphatidylserine, phosphatidylethanolamine. Membrane proteins also show orientation differences. What is the importance of asymmetry? A means without, symmetry means shape, without shape, functional specialization of leaflets. Signal transduction, negative charge is more inside, positive charge is more on the outside. Apoptosis signaling that is associated with phosphatidylserine exposure. Vesicle formation trafficking maintains polarity and stability of cells. Asymmetry in the fluid mosaic model. Membrane is dynamic and fluid. Proteins and lipids arranged asymmetrically, essential for communication, recognition, signaling, explain selective permeability and polarity. So here you can see sphingomyelins on the upper side, phosphatidylcholine on the upper side, phosphatidylserine on the lower side, phosphatidylethanolamine on the lower side, and in the purple color is the cholesterol embedded in the outer and inner leaflet. That is the outer leaflet and that is the inner leaflet. So here is again the composition of the outer leaflet and the inner leaflet. I have discussed it in the detail in the previous slide. Outer is the exophysial side and the inner is the site of facing the cell side. Cyto means cell side, exo means outside, facial means face. So these were the lipids and the structure fluidity of the membrane. Next is the membrane proteins. Two types of membrane proteins are present in the plasma membrane: integral proteins and peripheral proteins. Integral proteins extend through the lipid bilayer, are firmly embedded in it. Most integral proteins are transmembrane proteins. Peripheral proteins are not embedded in the plasma membrane. They are associated more loosely with the polar heads of the membrane lipids at the inner or outer surface of the membrane. Many membrane proteins are glycoproteins. So these are the membrane proteins we have discussing in the previous slide. So membrane proteins function includes ion channels formation, carrier formation, receptor action or enzyme action or cell identity markers such as glycoproteins and glycolipids. These identify the incoming molecules or signals. So third component was the structure of the plasma membrane was membrane carbohydrates. Carbohydrates are covalently bound to lipids to form glycolipids and to proteins form glycoproteins. These are mostly glucose, galactose, mannose, N-acetylglucosamine and N-acetylgalactosamine. Lipid raft is a dynamic, heterogeneous microdomain in the cell membrane, small structure. Microdomains means small structure that is enriched in certain lipids including sphingolipids and cholesterol. These microdomains are distinct from the surrounding membrane in terms of lipid composition and they play important role in various cellular processes. That is a lipid raft, a small microdomain inside the plasma membrane containing cholesterol and glycosphingolipids. What are the features of lipid rafts? Lipid composition, we first discussed it, that is the rich in sphingolipids and cholesterol. These lipids have saturated fatty acid chains and form more ordered tightly packed regions. Because they are not unsaturated, these are tightly packed, whereas unsaturated are loosely packed. If we talk about protein components, they are associated with the signaling, membrane trafficking, and cell adhesion. These proteins can be segregated into lipid rafts, including influencing their function, fluidity, and rigidity. Despite being more ordered, lipid rafts are still dynamic and can change their composition and organization. They can exist in both liquid ordered and liquid disordered phases, contributing the overall fluidity of the membrane. And if we talk about the role in membrane fluidity, they are also acting as a modulation. Lipid rafts can influence the fluidity of the membrane by creating regions with distinct lipid compositions. The presence of ordered lipid domains within the rafts affects the overall fluidity of the membrane, whereas they also contain the disordered phases as well, role in signaling. Lipid rafts are implicated in various signaling processes, like cell to cell communication is more important. If we talk about the prokaryotic cell, if we talk about the eukaryotic cell, all the cells are interlinked to somewhat to each other and they respond to each other. A cell is too much sensitive for the extracellular signaling as well as for the intracellular signaling. So next is the endocytosis and trafficking. Lipid rafts are involved in the membrane trafficking and endocytosis. They are acting as a sorting platforms for specific proteins, influencing their internalization and trafficking within the cell. If pathogen enters, what happens? Some pathogens exploit lipid rafts during the process of cellular entry because lipid rafts are the first they encounter while being chasing the cell. Lipid rafts can serve as entry points for viruses and bacteria, as these microdomains provide a favorable environment for the attachment and internalization of a pathogens. Internalization means to enter or reside inside the cell. They have the ability to invade these lipid rafts. Which have the ability? These are the microbes such as virus and bacteria. Pathogen means disease-causing organisms. Next is the cell adhesion. Lipid rafts are associated with cell adhesion molecules and play a role in cell adhesion and migration. They contribute to the formation of specialized membrane structures involved in these processes. And the last are the functions of the plasma membrane or cell membrane, transport across cell membrane. Transport of materials across the plasma membrane is essential to the life of the cell. It is also essential to maintain equilibrium of cell. Certain substances must move into the cell to support metabolic reaction. Other substances which are produced by the cell for export or as cellular waste products must move out of the cell. So transport is done through three passages: passive transport, active transport, bulk transport. We are going to discuss them one by one in detail. If we talk about the passive transport, simple diffusion, movement of particles from the area of higher concentration to the area of lower concentration down the concentration gradient, that is a natural process. It does not require energy and does not require membrane transfer proteins. Example: transport of gases, transport of ions, and transport of lipid soluble molecules. Next is facilitated diffusion. Diffusion of molecules down the concentration gradient, which requires carrier proteins. It does not require energy. It is more rapid than simple diffusion. It works as a ping pong. Absorption of fructose and glucose transport. Unipod antipod. Based on direction of movement of molecules, facilitated diffusion can occur by unipod antipod. Uni means single, transport of single type of molecules in one direction, such as example of absorption of fructose. Antipod, transport of molecule in opposite direction, e.g. chloride bicarbonate exchanger. Chloride on one side and bicarbonate on one side, opposite side exchanger. Osmosis, the diffusion of water through a semi-permeable membrane, movement of water molecule from an area of lower solute concentration to an area of higher solute concentration. Where solute is lower, water will be higher. Where is solute higher, water will be lower. So water is actually moving from its own higher concentration to the lower concentration. Although solute is lower, but water is higher. On the other side, solute is higher, but water is lower. So water is moving from its higher concentration to the lower concentration. But it is done through a semi-permeable membrane. That is why it is called osmosis. So decreased formation urine, hypovolemic condition, edema due to hypoalbuminemia, tonicity, and its effects on red blood cells. Active transport occurs through two passages: primary active transport, secondary active transport. In primary active transport, concentration gradient in which energy is used directly. It is carrier mediated. It requires energy in the form of ATP and used directly. Sodium potassium pump or calcium pump is the example. Also called as sodium pump, sodium potassium pump. In this pump, 3 sodium ions move out of the cell and 2 potassium ions move inside the cell with consumption of 1 ATP molecule. This is used to maintain the low intracellular sodium ion concentration and high intracellular potassium ion concentration. That is the sodium potassium pump. 2 sodium ions moving, the 2 potassium ions moving outside inside the cell, and 3 potassium sodium ions moving inside the cell. Next is secondary active transport. Transport against concentration gradient in which energy is used indirectly. The transport of two or three molecules are coupled. The transport is coupled with sodium potassium ATPase that requires the ATP. It occurs by symport and antiport. So symport is a co-transport. Molecules move in same direction, such as sodium glucose, sodium minus it is symport. If that does not occur, that there may be diseases such as cystinuria and Hartnup disease. And antiport is a counter-transport. Molecules move in opposite direction, such as sodium hydrogen exchanger that occurs in the renal tubule. And the second is the example of calcium hydrogen exchanger that occurs in the cardiac muscles. Next is bulk transport. Also known as transport by vesicle formation. It involves the formation of membrane-bound vesicles. It involves transport by macromolecules. Exocytosis. Exo means outside. Expulsing out of molecules from the cell. Fate of molecule released by exocytosis may be peripheral proteins, part of external matrix released to extracellular medium. Endocytosis. Engulfing large molecules by the cell. Two types. Phagocytosis. Large substances such as bacteria are taken within the cell. Pinocytosis. Fluid or fluid contents are taken within the cell. So that was the whole lecture about the plasma membrane.